then leads to Fanconi anemia. You also all know that there's a wide spectrum of symptoms in FA patients, but at the molecular level, all FA patients have the same inability to repair DNA crosslinks effectively. So if we look at the molecular defects in FA, there are over 20 different genes that can be mutated. And the FANC proteins encoded by these genes can be classified as components that act at 3 distinct layers in the pathway. So first, there's a group of proteins that associate with each other to form the FA core complex. And by far, most of the mutations are found in FA or in the FA core complex. Second, there are 2 proteins, FANCD2 and FANCI, that are the substrates of the FA core complex. And finally, the third set of proteins are involved in downstream repair. So, how does this pathway work? DNA crosslinks result in stalled DNA replication. This is sensed by the FANC proteins and causes activation of the FA core complex to add a ubiquitin protein onto its substrates, FANCI and FANCD2. Ubiquitin then acts as a flag to recruit downstream repair proteins to the crosslink. So these include nucleases that cut out and remove the damaged base, and proteins that actually repair the DNA. So a protein that conjugates ubiquitin onto other proteins is called an E3 ligase. And E3s work in a cascade of E1, E2, and E3 enzymes that transfer ubiquitin onto a lysine side chain of substrate protein. Question is, what are the roles that the purple proteins, FANCB, FANCL, and FAP100, form the catalytic core of the complex? However, the quantity and purity of this native complex were not sufficient for structural investigation. So we decided we needed to make a recombinant complex. And again, this work was started by Eason and would look like this in 2 dimensions. Now what we see is that the recombinant complex is of high quality and the structure is stable. Therefore, when we perform averaging, we can see these high resolution features in the complex. For example, the 2 lines circled here in yellow are 2 single long alpha helices. Now, using computational algorithms, we combined all the particles together to obtain a 3-dimensional representation of the FA core complex. And this is what it looks like. Overall, it's around 25 nanometers in length, and it has a fairly extended structure. At the center, you can see tube-like densities. These are alpha helices. As you move out towards the periphery, it's less well resolved than the central core. And we suspected that this was due to conformational flexibility. So the outside parts of the complex were moving around more than the inner parts. We could analyze this computationally and show that the FA core complex twists and bends. And this is what was causing the blurring at the periphery of the complex. Now, fortunately, we could resolve this by separating the 3D map into 3 separate parts. We separately analyzed the top, shown here in yellow, the middle, which is here in blue, and the base in pink. And now the map is much better resolved. All of these tube-like densities are alpha helices. So the map is now of sufficient resolution to dock existing structures and to resolve secondary structure elements. However, there weren't many existing structures of FA core complex subunits. They accounted for only 12% of the entire mass of the complex. So therefore, we needed additional information to figure out where each of the FANC proteins were located within this structure. And so I'm not going to go into great detail on this, but we determined where the subunits were located within the map using dropout experiments. We knew the subunits are colored in different colors. Now, interestingly, we found that there were 2 copies of the catalytic core, the FANCB, FANCL, and FAP100. These really sit at the center of the complex. The remainder of the subunits are arranged around this catalytic core. So we'll just look at that in a little bit more detail. So the FANCB and FAP100 subunits are shown here. And we see that they form not only the catalytic module, but they also form a molecular scaffold that coordinates the positioning of both FANCL, the ring subunit, and all the other FANC subunits. So here, FANC-B is in orange, and FAP100 is in yellow. And there's one FANC-B and one FAP100 in the bottom half, and one FANC-B and one FAP100 in the top half of the map. Now, I'd like to point out that a previous negative stain EM study from Alessandro Costa and Andrew Dean's labs suggested that in the absence of other subunit, this catalytic core really is a symmetric dimer of FANC-B, FANC-L, and FAP100. And one of the things we found really interesting was that even though these two subunits don't have any similarity to each other at the sequence level, they look almost identical in the structure. As I mentioned, there are two copies of FANC-L. The FANC-L has 3 different domains, and the model of FANC-L, based on a structure from Helen Walden's lab, including all 3 domains, fits into the base of the complex. And this FANC-L is really cradled by all the other subunits in the base. Now, in contrast, we could only visualize one domain in the second copy of FANC-L at the top. The ring was not visible, and we think this is because it's flexible and therefore blurred out in our structures. Now, because there were 2 copies of FANC-L and they had different structures, we hypothesized that they must play different roles in the. So here is what we learned from our structure using this cartoon representation. So FANC-B and FAP100 act as molecular scaffolds to coordinate the positioning of FANC-L and all the other proteins. Now, this explains why the intact complex is more active than isolated FANC-L, because it really stabilizes that FANC-L protein. Now, FANC-L in the base is fully ordered, surrounded by the other FANC subunits, whereas FANC-L in the top is flexible. We expect the green subunits, which are the substrate recognition module, to interact with the substrate proteins FANC-I and FANC-D2, and we hypothesize then that the FANC-L in the base plays a structural role to help scaffold the other subunits. On the other hand, the ring in the top FANC-L is accessible and it would be available to bind activated E2. So together, this would optimally position the substrate and the activated E2 to allow ubiquitin transfer. Now, this hypothesis has been confirmed by a more recent structure of the FA core complex, bound to E2 and substrate from the Pavlovich lab. So what does this tell us about disease? Most of the FA patient mutations are found in FANC-A, which is outlined here in blue. But it's in a dotted line for a reason, because we think that the FANC-A is flexible in our complex and therefore not very well ordered in our map. Now, some patient mutations are in the substrate recognition module in green in the base, but only a very small proportion are in the catalytic module containing the B, L, and 100 subunits. So I mapped the patient mutations onto the overall structure, and here the frequency of mutations are depicted by red dots. So you can see that there are many more mutations in the periphery of the complex than in the central core. Now, initially, when we first started studying this, we thought that mutations would really define the most critical parts of the complex and would tell us a lot about, by mapping the mutations, it would tell us a lot about how the complex worked. But this is an error, and we think this is an error of survivorship bias. And a classical example of survivorship bias is said to have occurred in World War II, when engineers were told to reinforce planes where they were hit by enemy fire. However, a statistician realized that damage on returning planes only showed the locations where the planes can sustain damage and still fly. Planes hit by enemy fire in other places do not survive. So in much the same way, our structures suggest that mutations are not well tolerated in the central core of the complex. And in agreement with this, the few patients who do have mutations in FANC-B, for example, have more severe disease, and cells lacking B, L, or 100 completely eliminate the ubiquitination activity. On the other hand, mutations in the periphery do not affect the structural integrity of the complex and are therefore tolerated. So overall, our structure of the FA core complex gave us insight into how this complex functions as an E3 ligase and provided insights into the mechanisms of disease. The next major question was, how is the FANC-D2 FANC-I complex ubiquitinated? And what exactly is the role of ubiquitination? On FANC-D2 and on FANC-I, as well as the regulatory, it's always difficult